Cramming for a test, trying to get more done than you have time to do. Stress is a feeling we all experience when you are challenged or overwhelmed. But then, just an emotion, stress is a hardwired physical response that travels throughout your entire body. The salivary enzyme alpha amylase has been proposed to indicate stress-reactive bodily changes in humans. Since it does not seem to be closely related to other biological stress markers such as catecholamines and cortisol, salivary alpha amylase may be a useful additional parameter for the measurement of stress. When stress increases, the production of salivary amylase increases. The higher concentration of enzyme, more starch is broken down to maltose and more maltose is produced. More maltose will lead to more enzyme produced, increases the stress level. Here, we conduct salivary amylase activity to test our stress marker among all our members group. The pre-stress absorbance values that show increment during the post-stress activities indicates that the salivary alpha amylase secreted into the saliva to digest starch also increases when the sympathetic nerve are activated during stress situation. The salivary alpha amylase of the saliva, that is one of the stress indicator, reflects the blood catecholamines which respond faster than the salivary cortisol, thus can effectively function as a stress marker. This is an enzyme. They are proteins that speed up chemical reactions. Without them, reactions can still occur, but they will be too slow to support life. Each enzyme has an active site. This is proof in the enzyme where the molecule that act on called substrate are captured and made undergo reaction. The substrate are either broken apart or combined together to form product. Enzymes are very specific. Each enzyme can only bind to one type of substrate. Let's look at the case of lipase enzyme, which break down to fats. Molecules like glucose and protein cannot fit into active site of lipase. Only fat cells can bind into active site because they have the right shape. This ability to act as fat cell is very important for lipase. The lipase convert fat into energy. What are the factors that affect enzyme activity? Well, we are focusing on substrate concentration and temperature effect using amylase. This is Michaelis-Menten graph. The graph shows relationship between the concentration of a substrate and the rate of the corresponding enzyme control reaction measured in velocity. There are two orders that present in the Michaelis-Menten graph, which are the first order and the zero order. The first order can be seen to occur when the reaction velocity is proportional to the substrate concentration, whereas the zero order happened when the reaction velocity is independent to the substrate concentration. There are two parameters that involved in the Michaelis-Menten graph, which is Vmax, the maximum rate at which the enzyme can catalyze the reaction, and Km, or Michaelis constant, which is the substrate concentration at half Vmax and provide a measure of affinity of an enzyme for its substrate. The effect of various temperature at 28 degrees Celsius and 40 degrees Celsius respectively on the enzymatic reactions are analyzed by using the line-Weaver-Berg plotted graph to find its Vmax and the Km value. The graph is obtained by plotting the reciprocal of V versus the reciprocal of substrate concentration. This shows then the curved graph of absorbance against the substrate's concentration.